And then share your screen. Yep. Yep, that green box. Who's coming in to do your online question?
I think Alana's gonna have her computer to like monitor the chat and everything.
Okay, when she comes in, just have her double check. Yeah, that sounds good. And then just a reminder that once you get going, you'll want to put on. Yeah. So good. And then did you decide is there any difference to the lights you want that I can deal with?
Honestly, I think this is fine.
I just leave it like this.
Yeah.
Okay. Sounds good. I have napkins and plates and stuff out there. Also, we'll be here in a half an hour. Okay. Weird to think that it's like the end potentially. And then, do you know what's next?
No, I did just sign a lease with my partner in Kansas City. I'll be there temporarily until we can both get jobs on the East Coast.
Okay. Is that where home?
That's where he is right now. He's a super in there. And then the East Coast, yeah, to be closer.
Okay. And then when do you think you're moving?
Hitting the road on June 30th. Is the goal.
Wow.
So once I do this, try to get rid of my furniture and apply for jobs.
Okay.
Yeah.
Just doing a full bite, going all the way.
I mean, we didn't anticipate signing the lease this soon, and we were like, we'll just do it.
A year lease?
9 months, because we don't really want to be there, but it
just works. You can
Yeah.
be spot.
It's like, it at least gives me the opportunity, if I get a job, to have a longer time frame wherever I am.
And his position is stable and long term.
Yeah, it's just the zoo field has such a low turnover rate, so for the positions he wants, it's few and far between.
So where he's at is where he's gonna improve it.
Until we can find something better.
What does he do? What does he work with?
He works with lots of animals. His main animal choice would be hookstock, like African hookstock, but he works with a number of leopard, gorillas, small primate, regular hogs.
Well, that's kind of
Yeah, I mean I think it's great. I get to go plant seeds when I'm there. It's very fun.
yeah, let's go play. And he comes to yours and he's like, I can't see it.
No, literally. But I feel like I have a bit more job flexibility than he does in terms of like leveraging skills, different things. Like I don't have to do science.
Crazy that you feel like you have the flexibility out of the two. Yeah, because usually it's not.
And honestly, I don't really know what I want to do anymore.
Okay.
In terms of like the current administration and everything, kind of thing.
I shifted.
Yeah, so it's like I might as well explore.
This is
the time. Like part of this chapter is trying something new. Kansas City has like bio type lab positions or working in university admin. So we'll see where I land.
And it's a braided path.
Exactly, it doesn't need to be there.
No, it so doesn't. That's an old school way of thinking.
Yeah.
Like at least for my parents. So like yeah, if I do this and I don't like it, I won't do it anymore. And I don't stuck on that career path forever.
Yeah. I feel like a lot of people in my generation are starting to lean that way. Right?
Let's just explore it. It just makes you a stronger human.
Yeah.
In whatever place you end up. So
exactly.
And then have you gotten all your writing stuff done?
Yeah, I just waiting for feedback from committee, I guess, after today. I think that feedback and edits I see fit. But otherwise, like it's all written. Well done. So just have to submit it to the grad school, and then I haven't taken the class right now.
Are you really?
Yeah, that was really dumb on my part. But
is it one that you wanted to take?
It's a class that I think will be beneficial.
Okay.
But I really don't want to be doing a final project on top of this right
now. Good morning.
If you don't mind hopping on with Zoom.
Yeah. I just want to double check everything looks and sounds okay. And then is Bob and Sue coming?
They should be. Sue told me she was gonna bring pizza for lunch, so I would like to think she's actually the show.
I was gonna start pestering about their cleanup plan for the labs.
Oh, I'm sure that's something you're thinking
It's on my
about.
mind. Oh, that's good, because man, there's a lot to be done.
I think they're planning on starting it too.
Okay, yeah, it's the hazmat stuff that I heard about,
Oh, with like the
all the
ethanol.
ethanol samples.
Yeah, we talked with EH&S about it.
Perfect.
When they came to do it.
Are they gonna require that you spit out the biologics from, or is it?
They just told us to like contact them when we do it, and then they'll help us.
We'll give it back.
Yeah.
The ideal would be, here is the container and all the stuff, and it's yours.
Yeah, they're also working on trying to see if they can find people to take the samples,
Okay,
so they don't have to be thrown away, but
Yeah.
that's obviously a great part, because they're all involved transporting ethanol.
right, right, and getting somebody who wants, right, and needs the samples as well, and house them.
Yeah.
Pretty crazy to think about that all came on that.
Yeah.
They've both been here longer than me, so.
Okay, you want me to start this? Join it.
It's just join
Alright.
it. If it's not, then let us know.
Yeah.
Join.
I should have given Teresa like an alternate email. Like also.
And then,
Yep, it's working.
are you a co-host?
Yes.
Okay. So in there, let's look at it. Look at it. For whether or not there should be if anybody.
Oh yeah, go ahead.
Okay, so I would actually turn that one off, do that one on, for that. And possibly making this one off. That's kind of up to you, but typically I don't let order in defense. They can only type.
Yeah.
Because then they kind of just ask very long questions, and this way it allows you to curate.
Yeah.
Okay, so when the Q and A's happen, then it should say, please type your questions into the chat, do not turn on the mic. Or you
No,
can't turn on your
we're not
mic.
letting them.
We took like a pause.
Awesome. That's great.
Okay, great.
Okay, and then. That, you should see everybody. And so if there's anything weird, if they're turned off, do them because you're opposed to all that. The other thing I watch for is that if anybody has an AI bot, I can get
Ah. Does it say like AI on there? Okay, cool.
I
I am happy to kick out some AI. Yes, that sounds fun.
So it goes in and it basically takes audio, it listens to the audio and makes written notes. And you haven't given them permission to do that, and so I can. Because you haven't published yet, you haven't done this kind of stuff, and so that's, I'm just, I really watch it.
Yeah.
Yeah, so it depends on how the Zooms are set up. I actually, on my side, before I set it up, I can make sure that you can't do that. But if I don't know how this will set up, just watch it.
Okay.
And then if you just hover over their name, the little, you can like, click on that, it'll say, you are a host.
Yeah, so I can't
Yeah,
everybody,
everybody as a participant, well, that's a panelist, but
Okay.
these.
And
So it'll be there, and then you can just check, but because it's a Zoom or a webinar, they should.
Okay.
Yeah, I turn my mic on, can you see the process? Okay.
Hello. Hello.
You're all good.
And do you have any remote people on your panel, your committee?
I do think that Francis, I choose for her.
Okay, is there any reason they have to be able to? During this part?
Not during this part, I have the separate Zoom.
Yeah, so that would be the only thing, because they'll come in, and if they want to.
Okay.
And then you talk.
If they're like, I'm on the committee, I wanna talk. Okay.
Right.
I
And you
will-
say, yes, of course. Anything you need.
Actually, you're gonna question me for an hour after, don't.
Right.
Yeah.
Okay.
Yeah. I mean, what else am I gonna do? Hang out at home?
No, yeah. Okay. Want me to come back later?
What? I think everything's good.
I can just-
I already, you know, just in a nervous bathroom break and
There'll be like 2 more
before
before
it
that.
starts.
That'll be fun.
Is this fine for my?
It sounded just fine. And you can always do just the sound check. Say, can you hear me? Can you hear the audio? Okay, let us know if you can't hear anything right now.
I can also like go up there, the computer, so it doesn't have feedback that way.
I'm sure I will get bombarded if people join early and they'll be like, it's all working.
Oh, great, that
I'm
like
sure
sure.
question is facing. I'll be in the room too, so if you want to stay here.
Okay.
But yeah.
Meredith, any questions you don't want me to like ask?
Good question.
I know it's would you be.
Yeah, like because I can choose the question.
Oh,
So, or any people you don't want questions from.
if you see anyone named Rachel, don't Rachel Goldblum, if she's talking on the Zoom, don't let her ask questions. That's my old
Is your mic off?
Yes. I mean, I think she didn't like, but I like to think she wouldn't be on the Zoom.
Oh, your roommate from Corvallis?
Yeah.
Oh, yeah. It was probably.
Okay, man, the only other thing I would suggest in this stuff to you is not to be able to see that.
Okay.
The reason is that you're gonna see how many participants and you're gonna see every chat that comes in and it's gonna distract. But if you let it, you or not.
Oh,
It goes-
so I can just hide it. You go Amy's app.
I should. Yeah, and if you ever need them back. You can actually have that if you want it. You wanna hide that?
I mean, it doesn't really bother me.
Okay.
Yep. Perfect. Okay.
I'll be back as we get closer.
So exciting.
Go to those.
I like your outfit. I like the coordination of the brown Converse with the brown shirt.
I originally was wearing a tank top and I was like, the push bitch section.
Depends. No, it's gonna be great. It's gonna.
It's just y'all.
Good. Shut the food.
I think Cinnamon's gonna have coffee in that little lobby area.
In this area?
No, like I think off this. Or is. She's really. Well, the polls, I feel like it's a little than like a proposal meeting.
Yeah.
You don't think with a webinar, you have to let people in? Whoa, there's somebody on. But, sure, but then- Cheers. Okay, you should know.
Oh,
And she made me like actually, I just.
Yeah.
oh, I would go mute you for a moment.
Oh, true. I can also just text her.
We said mute. great scientist and that's been, you know.
Yeah, for some reason,
Well, the
percent optics not working. So my slides better name work.
Okay.
It should work.
Okay, I'm not touching it, but now there is a magic, that's you got set.
Yeah, there.
Interested in it.
This doesn't work.
Oh, even if it's a little more maybe.
Right, I'll stop this and do this.
That'd be great.
And toss it at the same time.
Vibration.
And walk and chew gum.
Hey, folks.
It's the main thing.
Exactly. Super dynamic, very.
I was telling me this new thing that
You
you learned about keeping your brain active is, you know, when you go like this,
Yeah.
do one opposite on each hand.
Oh.
And like I tried to do it on the drive in today, wasn't happening. I was clearly just too distracted. But so yeah, start with,
Trying to think.
yeah, you're doing, it's making both sides of your brain work. Working on.
Yeah. Well, there we go.
Now I got it.
Hard.
Both sides are working.
Yeah.
I rebooted them.
Okay. Start talking to you.
All right.
Do something or something else.
Yes.
You know why you got this hard.
You are in mute now.
Yes, everyone keeps
But
it.
like you're.
It should be.
It should.
Action it.
Nothing new, but like. So, see it. Yeah.
Well, those are.
Elena. Or you. If, can you hear me on there?
Stop there.
Almost, oh my gosh.
Hello. Okay.
I don't have
The speaker.
It's a sound right. Use this just for the online.
Yeah.
Not. Okay. Whoops. I have to talk and chew gum here. I have to do this and speak coherently. So we'll give this a try. I think you all know who I am. Myself, but it's my pleasure to introduce Meredith Anderson today for her master's defense. Excuse me. Meredith came, I don't know, maybe about 3 weeks ago with a new title to her thesis, Zoeal Odyssey, and I thought, Odyssey, let's explore that a little bit. The first thing that came to mind was Homer's Odyssey. So I was, that epic poem that we once knew a long time ago, probably. Now, I don't think she's going to do her defense in verse today, but I do think it warrants, she was on a journey. And so I do think Odyssey is appropriate. In fact, there was ships involved, there were some waves, various adventures. And if you look real closely, you can even see Meredith on one of these ships. But I thought instead of wandering through the Mediterranean to give her background, that I'd start with her actual diagram that she created and give you a little sense of what she was doing prior to this. So I don't have any goofy pictures of little Meredith. She hasn't been here long enough to dig those up. But she spent time, grew up down south, far, far away from here, and did her BS at University of Florida, where she earned a number of accolades and awards, including the Medal of Excellence and Outstanding Marine Sciences Student and Top 10 Senior, among others. While she was there, she also worked for the USGS as a researcher and student contractor. And she also earned the prestigious Noah Holling Scholarship, where she worked the Northwest Fisheries Science Center in Seattle. And she worked on warming and ocean acidification effects on the early life stages of Pacific krill, which set her up beautifully to come to OSU, where she joined the Department of Integrative Biology in 2024. She spent a year working in a TA ship and went out to sea, as you can see in the photo photo up there. She also presented her work in second place recently at a presentation. She volunteered both, I won't read through this, but both in her home department, Integrative Biology, as well as out here at Hatfield. But mostly she spent a lot of time sorting little tiny spiky things out of the plankton. And she worked very diligently, very quickly, and at this point has already submitted 2 manuscripts for publication, which is truly amazing. And she's going to talk to you about that work today before she goes off and leaves us for other journeys and adventures. I just thought I would, before she gets into it, show you a couple pictures by Mark Farley, just showing what Meredith looked at for hours and hours and hours. When you start to see some of the numbers she's talking about, you can think of these little tiny spiky things, which I often think of as these little burrs that we all get on our feet if we walk barefoot outside here. But I will pass it over to Meredith now. She doesn't need to hold this thing. Turn this off.
All right.
Give this to you.
Thank you,
And go
Sue.
sit down. Take it away.
Thank you, Sue, for that introduction. Thank you for everyone who's here in person and online. I'm excited to share the work of my master's thesis with you all that looked at the spatiotemporal distributions of Dungeness crabzoea in the Northern California current. And so to give a brief overview of how the presentation will go today, my thesis covered 2 chapters. The second looking at the temporal and spatial zoeal distributions along the NCC, and then my following chapter looking at the fine-scale oceanographic drivers of those patterns observed in chapter 2. And because these 2 chapters are largely complementary, I plan on talking about them together rather than as 2 separate sections. So to begin, as a brief overview, those of us on the West Coast are likely familiar with Dungeness crab in terms of its fishery context. We may see headlines such as these in our day-to-day regarding season delays, changes to gear restrictions. And while I'm unfortunately allergic to my study organism, I have been told it's quite delicious. And so historically, the fishery started in the 1850s in California, but I'd like to acknowledge that this organism has been traditionally harvested by tribal nations all along these coastlines long before the 1850s. When we think of the present day fishery, it's managed under a 3S framework, size, sex, and season, essentially meaning we can only harvest males of a minimum carapace width during the specific open harvest season. And as you can see in this graph, it is a multi-million-dollar fishery on the West Coast. But across the different states it supports, California, Oregon, and Washington shown here, it exhibits pronounced interannual and regional variability. But of course, this fishery does extend into Alaska and Canada, but for today's talk, I'm only focusing on these regions. For those of us, when we think about Dungeness crab and crab in general, we usually think of the adult stages. Those are benthic organisms, meaning they're at the bottom of the water column. But for a lot of benthic marine organisms, they exhibit a biphasic life history where their larval stages occupy a habitat distinct from the adult stages. For Dungeness crab, their larval stages occupy the pelagic environment in the open ocean at the will of the currents and waves. For our region, Dungeness crab typically extrude their eggs in the fall. Those eggs then hatch into the zoeal stages, which go through 5 zoeal stages in the winter months. Those final zoeal stage metamorphoses into the megalopal stage in the spring and summer. And megalopy then return shoreward to our coastlines, starting the benthic life cycle for Dungeness crab. And why should we care about the larval stages, you might ask, when we think about a fishery species that we're harvesting the adult stages of? Understanding the early life history dynamics can be really critical because these can provide information as to why we might see recruitment variability in any given year. So understanding what can drive different mortality rates in the larval phases, understanding how these larvae are distributed across space and where they might end up can all help fishery managers in recruitment modeling understand why fishery yields might be bad one year and good another. For Dungeness crab, a lot of this work that ties the larval stage to the adult fishery context has focused on the megalopal stage. Alan Shanks and many researchers have used light traps to collect nearshore returning megalopy, and they found a pretty strong relationship between the number of megalopy caught along the coastline, typically at Coos Bay, to commercial crab catch lagged 4 years later. But we still see unexplained variability in the number of recruiting megalopy. And so while these light traps have provided a convenient way to collect these megalopy, and sometimes they do catch zoea, we still know very little about the offshore variability that these zoea are exposed to. And a lot of that has to do with offshore sampling in the winter along our coastlines is dangerous. We're often limited in being able to get into the field due to winter storms. For those of us that go out into the field in the winter, you likely know. The historic research around Dungeness crab has largely been limited in its sampling locations, whether along Newport Hydrographic Line or at Coos Bay. And the foundational historic research that does look at Dungeness crabzoea has largely been done 40 to 30 years ago. And so when we think about the modern contemporary work that has looked at Dungeness crabzoea, it's not necessarily focusing on this species as the sole species of interest, but is instead considering all species of different crabzoea. And this work has been predominantly focused off of Bodega Bay and Point Reyes. And this is associated with a headland which alters circulation patterns and thereby influence zoeal distributions differently than what we might see here. So we're not necessarily able to extrapolate those findings from more modern research to our own coastlines. And lastly, the research that has been done is using Tucker trawls, bongo nets, and so on. And the goal of my thesis are to improve our understanding of the variability we see in these zoeal populations across the NCC by providing 4 years of winter data across a broader portion of the Northern California current in the modern context and using high resolution depth discrete sampling methodologies in the offshore. The objectives of my thesis were to characterize the temporal and spatial offshore distribution and abundance patterns of Dungeness crabzoea. My second objective was to then investigate the fine scale drivers of those patterns. And then using this novel methodology, looking at what are these zoea experiencing in their environment in situ, and also looking at what plankton community members are they most often co-occurring with. So how do we collect this data? Well, we sampled this portion of the NCC as shown in our sampling map during the winter months of February and March. And so we sample along cross-shelf transects, which are represented by the lines on this map, and at predefined stations along those transects. We sampled in the winters of 2018 and 2019 during the mezcal cruises, which are colored in blue on the map, and during 2022 and 2023 during our spectra cruises, which are colored in orange on the map. As you can see, mezcal sampled 2 transects, one in Northern California at Trinidad Head, and one in Central Oregon, Newport Hydrographic Line. Spectra, on the other hand, those cruises sampled from Central Oregon at Heceta Head all the way to Southern Washington. To sample these transects, we use a complementary approach where we have biological net sampling and plankton imaging. To collect our biological net samples, we use MOC-NES, which is the multiple opening closing net and environmental sensing system, which is a mouthful and why we call it MOC-NES. This is an offshoot of the Tucker trawl system. And MOC-NES allows us to get depth discrete sampling intervals because we can deploy this coupled net system at 25-meter depth intervals to a maximum depth of 100 meters at our predefined stations. Our lab uses a coupled MOC-NES system, which means we have 10 nets that we tow behind the boat. 5 of those nets have a larger mouth size and larger micron mesh, as shown in this bottom right blue diagram. And 5 of the other nets are a smaller mouth size with finer micron mesh. And for the purpose of my thesis, I'm only working with samples from our MOC-1, the smaller net size. In addition to our biological net sampling, we also complement it with the in situ ichthyoplankton imaging system, otherwise known as ISIS. This is a high-resolution shadowgraph imaging system that we tow continuously behind the boat in a tow-yo pattern along the entire length of the transect. This allows us to quantify taxa that has often been under-resolved in plankton net sampling, such as gelatinous taxa, and also allows us to have an even finer scale resolution at 1 meter depth bins compared to the 25-meter depth bins that MOC-NES provides. ISIS is also fit with several environmental sensors and probes, and it allows us to simultaneously collect physical data alongside our plankton imagery. We take these samples back to the lab, where, as Sue mentioned, I sat at a microscope for hours and picked out all of the crabzoea and megalopi. Using dichotomous keys, I then identified those zoea and megalopi to the lowest possible taxonomic level, which was often to the family level. And then for anything I identified as Dungeness crab, I staged those zoea from 1 through 5. How do you stage a zoea? Here we have a schematic of a late-stage Cancridae crab zoea. Dungeness crab belong to the Cancridae family. In the presence, absence, quantity, and length of the certain features that are labeled on the schematic can be indicative of the zoeal stage. The reason I said this was a late-stage Cancrid crab zoea is we see the presence of pleopod buds along the abdomen. We can count 10 swimming setae and 5 inner setae, But of course, not all crabzoea that we sample have this morphology. We also have a diversity of other taxa that we sample with our mock nest system that we identify, and we can see that their body shapes and body plans can be quite different, including the porcelain crab in this bottom right corner, or bottom right corner, the pea crab in the top left corner, and the mole crab in the middle. In addition to the lab processing, we also have to process the thousands and thousands and thousands of data points that ICES provides. This is a very complex process, so I'm only going to give a brief overview of how we process this data. But because of the large amount of data that ICES provides, we use an automated machine learning pipeline to efficiently classify all of this plankton data, where we take the continuous raw ICES images and segment them into regions of interest, as shown in these smaller plankton individuals. Those segments then get fed into a classification model where a training library of 170 or so taxa classes get mapped onto those segments, essentially saying this pink square is a crustacean crabzoea, this yellow square is an amphipod, and so on. But with any automated machine learning pipeline, we have to account for error, we have to validate the work that's being done. And so we take a test set of that classified data to create probability thresholds to then adjust these classifications and filter low probability images. This final classified data frame then gets merged with the simultaneously collected ship and sensor data into a final data frame where we have binned abundances per 1 meter depth bins. But as you can see in this pink square, this pipeline cannot resolve the species or stage of the crabzoea. And that is when the mock nest sampling becomes really important in being able to use this data. In my mock nest data, I essentially had proportions of Dungeness crab at each transect station and depth bin. And using the nearest neighbor matching method, I then matched the most spatially proximal Dungeness crab proportion from our mock nest data within the binned abundance data, adjusting those proportions to account for the fact that not all crabzoea that we sample with our mock nest net is a Dungeness crabzoea. We have to account for the diversity of other crabs we're collecting, both in our images and our net sampling. Once we process all this data, we're able to visualize it. And so here we can actually plot our raw data and see the continuous toyo path that ICES took along the length of the transect, in this case, Columbia River. To orient you, our shore would be on the left side of this graph. And these dots are colored according to the calculated binned abundance of overall Dungeness crabzoea. And as you can see, there's a lot of dark purple, a lot of darker blues indicating lower abundance, which is typical. But you might be able to visualize and see some spots where we might see higher abundance with some lighter yellows and greens. But as a whole, showing our data like this is not very digestible, it's not very interpretable. So we're able to then spatially interpolate our raw data onto a two-dimensional grid where we have depth on our vertical axis and relative distance on our horizontal axis, where we have the dashed line at 0 representing our shelf break. This dark gray polygon in the left corner represents the continental shelf that we sampled over. And here we can see these Dungeness crabzoeal abundances are visualized in a much more digestible and interpretable way. And we can see that they can be incredibly patchy in a way that we would not be able to resolve with net sampling alone. So imagery data is allowing us to see these distributions at a much finer scale. To analyze this data, for my first objective in looking at the broad scale and temporal and spatial patterns in these zoeal abundance data, this is when I'm using the mock nest data only. I utilize generalized additive models, or GAMs, with a Tweedy error distribution to account for the fact there are a high presence of zeros in plankton data like this. And I use generalized additive models looking at these patterns at the Newport hydrographic line, the transect we sampled across all 4 years, as well as spatial models dedicated to the 4 cross-shelf transects we sampled during these spectra cruises. I also assessed vertical positioning in the water column using mock nest provided weighted mean depths and analyzed the differences between weighted mean depths across the wheel stages and during our day-night sampling. And I did this using non-parametric Kruskal-Wallis and paired Wilcoxon signed rank tests. To then move into my second objective of investigating the fine scale drivers of these zoeal distributions, this is when I started using the mock nest ICES integrated data set. This is a lot more data than the MOCNIS data alone. And because of that, traditional statistical methods like generalized additive models are computationally inefficient in being able to analyze this size of data. Because of that, I used a non-parametric ensemble machine learning technique known as random forest modeling to, one, be able to analyze this very large data set efficiently, but also still be robust to collinearity that we also see in plankton data like this, be robust to zero inflation and noise in our data. I used random forest modeling to look at the influence of spatial predictors, depth and distance from shore, as well as a suite of environmental predictors, including temperature, salinity, pH, dissolved oxygen, and chlorophyll A. Using this integrated data set, I also looked at environmental exposure using linear mixed models, as well as plankton community overlap patterns with Spearman's rank correlation analysis. So, of the 290 MOCNIS jars that I sorted, I identified 153,032 zoea. It's a lot of zoea, a lot of tiny guys and staring at a microscope for many hours. How many of that was Dungeness crab? 48.9%. So on average, 74,760 of that total. And that's to say this is 48.9% across the board of the 4 years of MOCNIS sampling. But we did see variability in those proportions across the different transects, stations, and depth bins. Here we have the overall concentration of Dungeness crab across the 4 sampling years of MOCNIS along the NCC. And we can see that there is variation in the magnitude of concentrations across the given years. We can see in 2018, 2019, and 2023, Dungeness crab zoea are predominantly in the inshore. We can see some areas where their concentrations are much higher. But in 2022, we can see these distributions are more spread out across the shelf. So when we are sampling and we see this level of temporal and spatial variability in these Dungeness crab concentrations, we have to consider spawning phenology, where and when these Dungeness crabs spawn in relation to when we sampled. You know, we're sampling 1 week of a given month during these cruises. What are we actually catching in terms of when these crabs are spawning? So we could be catching one spawning event in our sampling. We could be catching continuous spawning events, which could explain why we might see more broad distributions across the shelf in 1 year versus the others. We also have to consider the fact that we're also capturing different source populations of Dungeness crab. And all of these things could influence the temporal and spatial variability that we observe in our MOCNIS catch. Overall, we saw that there was high interannual variability in zoeal abundances. Years in our GAM was nearly always showing a significant correlation. Here we have a bar plot of the overall concentration of Dungeness crab along the transect we sampled across all 4 years, Newport hydrographic line. And we can see that these concentrations can vary by several orders of magnitude in any given year. And we found in our data that winter 2023 was significantly higher than our other years sampled in all of our GAMs pretty much. And this could be due not only to spawning phenology, but we also have to consider the variability of the oceanographic context these zoea are within. I'll show some more environmental data further on into my talk, but winter 18 and 19 were our warmer years on average. In 2022 and 2023 were cooler years on average. These can influence development rates. They could also cue different spawning timelines as well. And so because of that, we're seeing different environmental characteristics during these spawning timelines, which could influence the temporal variability we observe. We also have to consider broad-scale climatic cycles, such as Pacific Decadal Oscillation, or PDO. This is a phenomenon that influences the Northern Pacific in terms of sea surface temperature, upwelling intensity, current strength, and so on. And research has found a positive correlation when PDO is in its negative phase in the number of returning megalopy. So we had assumed we would see higher zoeal abundances during negative PDO phases, but all 4 years that we sampled were during negative PDO phases. So we were not able to attribute these broad-scale climatic patterns alone to the variability we're seeing here. But all in all, the variability we observe across these given years in zoeal abundance do reflect the variability we see in the number of returning megalopy and could indicate that recruitment variability may originate in the offshore. We also found that the cross-shelf position of the zoea is structured by ontogeny, or the individual stage in development. Here we have the panels broken down by individual zoeal stage and their concentrations across the station sampled with mock nests, with station 1 being our most inshore station, and station 6 on the left being our most offshore station. The pattern we observed was that our early stage zoea, zoea 1 and zoea 2, were predominantly in our inshore stations. Zoea 3, we're in the inshore, but we're starting to occupy more stations across the shelf. And then zoea 4 and 5, our late stage zoea, were predominantly found in offshore stations. This is consistent with historical observations of Dungeness crab zoea in the region and other regions as well, and this is likely reflecting spawning phenology. So these early stage zoea are likely to be found closer to where they respond, whereas the further you are along in zoeal development, the longer you are exposed to cumulative cross-shelf transport, which is why we might see these late stage zoea are found further offshore due to their development timelines. A commonly cited reason for why we also might see cross-shelf adjustments is larval swimming behavior in vertical swimming. These zoea cannot necessarily adjust their horizontal position, but they can adjust their vertical position within the water column and potentially exploit different depth-varying currents. And so the idea is if our late-stage zoea have a relatively more enhanced swimming capacity, they would be able to occupy different areas throughout the water column and exploit more depth-varying currents, thus progressing further offshore. But for reasons I'll show later, we did not see evidence of this behavioral cue in our late-stage zoea. So again, this likely reflects cumulative offshore transport the longer you are in development across ontogeny. We found that this pattern generally held true across the NCC. This is our GAM model output for our stage-specific GAMs, where we have the distance from shore by transect with Grace Harbor on the top and Heceta Head on the bottom, and then each column is the stage-specific GAM. We did combine zoea 4 and 5 into a late-stage category to account for the number of zeros in the zoea 5 category. And our model outputs generally showed that predicted concentrations showed peaks in the inshore for our early stage zoea 1 and 2. Zoea 3 exhibited more spatial variability in where they were across the shelf across any given transect. And late stage predicted abundances generally showed movement further offshore. Distance from shore was often significant in our GAMs, but of course we did see variability in that Columbia River, zoea 2 and zoea 3 models showed non-significance as well as in our late stage GAMs at Newport Hydrographic and Heceta Head. We also found that zoea were predominantly concentrated in the upper 50 meters of the water column. Here we have the relative density of zoea across the individual zoeal stages with depth on the vertical axis, and the average weighted mean depths are reported in the left bottom corner. We found that depth was always a significant predictor in our GAMs, and the relationship between the abundance of zoea and depth was roughly linear. We found with our weighted mean depths that the average depth occupied throughout the water column by these zoeal stages did not vary across ontogeny, further providing evidence that we did not see these late stage zoea occupying different areas of the water column to potentially exploit depth varying currents. In addition to this, we did not find evidence of diel vertical migration, or DVM. DVM is a phenomenon we often see and observe in the ocean where organisms will migrate to the surface at night to forage and then migrate to depth during the day to avoid predation. However, while we did not see evidence of DVM, we are seeing something happening at night. With the exception of zoea 1, we do see these zoea are occupying a broader portion of the water column during the night than they are during the day. So potentially with less visual predation at night, these zoea can occupy a broader portion of the water column to have improved foraging success and then constrain their distributions during the day under more visual predation potentially and of improving potential survival outcomes. In summary, our mock nest data shows that we saw high interannual variability in these zoeal abundances across the years. We saw that zoea were structured across the shelf through development. They were largely in the upper 50 meters of the water column, and there was no evidence of DVM. But what does that mean when we look at these distributions at a much finer scale? What does our imaging resolution provide that we could not resolve with our mock nest data? And we found in our imaging that the Dungeness crab zoea were incredibly patchy across the shelf and with depth. Here we have our spatial interpolations of overall Dungeness crab abundance in winter 2022, the top row, and winter 2023 on our bottom row, across the transects sampled, which are our individual columns in this panel. And so first, we can see that the magnitude of difference of zoeal abundance is reflected across the years. We can see the interannual variability. We can also observe that when we sampled along the length of the transect with the presence of the 200-meter shelf break, shown by this white dashed line, generally, we saw that these Dungeness crab zoea were predominantly found before the shelf break. And we saw transect level differences. Most notably, when we look at Columbia River in 2022 and then Heceta Head in 2023, we can see that these Dungeness crab zoeal distributions are showing roughly similar patch structure. And this is important because Columbia River and Heceta Head are both transects that we sample that are associated with retentive features. Columbia River has the influence of the Columbia River plume, which interacts with the ocean front and is known to retain nutrients, organisms at that convergent zone. And then Heceta Head is associated with Heceta Bank and broader continental shelves, which are known to alter local circulation patterns and have been shown to retain larvae in this region. And so this is notable because we are seeing transect level differences, but we do see similarities at transects with similar retentive features. So when we actually then are able to look at these fine scale distributions, what does that mean for the predictors and how they might manifest across these distributions? When we looked at our random forest modeling, we were able to visualize the relative importance of the predictors included in each model, where we have our predictors reported on the vertical axis and our individual models reported on the horizontal axis. This is a heat map of the relative importance of an individual predictor, with the darker colors representing higher importance and the light yellow colors are representing lower importance. The predictors that are showing white are predictors that were not included in our model. Generally, our random forest modeling, we're able to explain a low to moderate proportion of the variability we saw in these abundances, ranging anywhere from 35 to 65% variation explained. And across our models, we saw that distance from shore was nearly always the most important predictor across all models, with the exception of zoea 2 having chlorophyll A rank marginally higher. This is important because while this is ranking as the most important predictor across these individual models, that's not to say that its influence is the same, and I'll show some examples of that. Of the environmental predictors that we included in our random forest modeling, chlorophyll A concentration was surprisingly the most important. This could be indicative that these zoeal stages need to occupy areas across the shelf and with depth that are relatively more productive in the winter. And these chlorophyll A concentrations are likely indicating trophic structure in areas where these crab zoea can forage. And so we know that for the early life stages of many larval organisms, first feeding can be a bottleneck for larval survival and development into the next stage. So the importance of chlorophyll A here could be indicative of these early stages that we need to be able to be in a region where they can feed and forage and be able to develop into the next stage and survive. If we move past distance from shore and chlorophyll A and just focus on the left side of this heat map for our stage-specific models, we can see that there are differences in the predictor importance. For instance, pH ranks the third most important in our zoea 2 model, but we do not see that importance carried over across ontogeny. This is important because these zoea may be occupying different water masses, and because of that, the environmental characteristics of the water masses that they occupy could manifest differently in how these predictors influence their distribution patterns. And lastly, time of day and depth exhibited negligible importance in our models. And so we're able to visualize the effect of a predictor from our random forest modeling using accumulated local effect plots, which is looking at the local effect of a predictor as reported on the horizontal axis here, on the predicted response. In this case, the predicted zoeal abundance of Dungeness crab. And ale plots essentially show us that anything above the 0 dashed line is a positive deviation from the mean of the predicted response, and anything below the 0 dashed line is indicative of a negative deviation from the mean predicted response. And so these are the top 4 predictors from our overall zoeal models, with spectra model on the top and our mezcal model on the bottom. And distance from shore, again, ranked the first most important, but we can see that the relationship with distance from shore is varied across both models, where with spectra, we can see that predicted abundances are expected to increase in the first 45 kilometers, and then we see a negative deviation as you move further offshore. Whereas in our mezcal model, we can see that the predicted abundance is predicted to be higher in the first 25 kilometers, and we see a much more noisy relationship as you move offshore. We can also see that in the shared fourth most important predictor, dissolved oxygen, we also can exhibit differential impacts of a predictor on these abundance patterns, further indicating that we do see environmental variability across the years we sample, and that might manifest differently in how these abundances are structured across the shelf and with depth. For our spectrum model, chlorophyll A ranked as the second most important, which also ranked as usually the second most important in our stage-specific models. And the relationship shown here is generally what we see in our stage-specific models, where abundances are predicted to increase beyond 2 micrograms per liter and decrease at values below that. And then with mezcal, we see that predicted abundances are expected to have a positive deviation at temperatures below 10 degrees Celsius, and then negative deviations as you move beyond 10. This is notable because mezcal were our warmer years on average, but we do see a similar relationship in our spectra overall in stage-specific models in terms of temperature. But we then are able to tease these relationships apart even more when we then look at the individual zoeal stages. And so here we're looking at the effective distance from shore on the across ontogeny, and we can see that the relationship shown in our overall model gets broken down even more so in a way that is consistent with what we saw not only in our mock test data, but is consistent with what we see in our historical observations, where our early stage zoea show positive deviations in the first 50 to 25 kilometers, and then we see negative predicted abundances as you move further offshore. With our zoea 3 model, we see that these zoea are predominantly showing predicted higher abundances in the first 25 kilometers, and then a more variable relationship as you move across the shelf. And then zoea 4 and 5 are showing the inverse, where we see positive predicted abundances as you move further offshore. But what's exciting about this is then we're able to see the reflection of our random forest results in our spatial interpolations. So what we're seeing here is the spatial interpolations of the individual zoeal stage distributions across the Newport hydrographic line. And so here we can see the reflection of the relationship in our random forest modeling in the actual data. Our models are only as good as our data can provide, and we can see this reflection here, which is really exciting. And so generally we're seeing this offshore progression as these zoea are developing through time. This is important because distance from shore isn't necessarily a causative effect, but it's probably integrating multiple different physical and environmental gradients across the shelf. And because of that, when we're able to look at a much finer scale resolution as we're moving across the shelf with these zoea, this is likely impacting the environmental conditions they experience. Here, we are looking at what is the general exposure that these individual zoeal stages experience across ontogeny with any given environmental predictor. So each column you see here is the environmental parameter that we looked at: temperature, salinity, chlorophyll A, dissolved oxygen, and pH across the individual stages with zoea 1 and zoea 5. And essentially what this is showing is what is the distribution of any individual zoeal stage across the range of the environment. And to draw your attention first to temperature and salinity, we can see that zoea are experiencing conditions in situ in their environment that are not matching what we use in terms of optimal larval development in our experiments. Often, temperature is cited as being 10 to 14 degrees Celsius for optimal larval development, but our spectrum years that we sampled, these zoea are generally experiencing temperature conditions that are lower than 10 degrees Celsius. In our Mescal years, those were our warmer years on average, and they are showing zoea occupying anywhere from 10 to 11 degrees Celsius. But our models did show that we saw higher predicted abundances at lower temperatures. For salinity, 25 to 30 is often cited as the optimal range, but across all 4 years, we generally saw that all zoeal stages are experiencing salinities much greater than 30. This is important because when we consider what are these zoea experiencing in the wild versus what they might experience in the future under climate change, this might suggest we need to reparameterize how we adjust our experiments according to ambient temperatures, as well as potentially reparameterize models to consider what are these zoea experiencing in situ and how will these conditions then change under future ocean regimes. And these can have differential impacts on larval survival, most notably with temperature. Warmer temperatures can induce quicker development rates, produce smaller larvae, and increase mortality. And lower temperatures would show a slower development rate, faster larvae, or larger larvae, and higher survival typically. So these could have differential impacts as well on the development outcomes of these zoea. We also saw that these early stage zoea, the first 2 rows, are often experiencing a broader range of environmental conditions than their late stage counterparts. To draw your attention to dissolved oxygen in pH, we can see that these early stage zoea are experiencing both low and high dissolved oxygen in pH conditions, whereas their late stage counterparts are experiencing the upper thresholds of the environmental parameter that we sampled. This is important because we have to consider where these zoea are across the shelf. With our early stage zoea being predominantly in the near shore, they are experiencing broader environmental conditions. And because of that, this could have differential impacts on their survival as these conditions continue to change under future climate scenarios. As we move further offshore, these late stage zoea are occupying arguably more homogeneous water masses due to winter mixing and turbulence, potentially causing these environmental conditions to be more constrained. Then when we move into, okay, where are these zoea across the shelf? How might that not only influence the environmental conditions they experience, but what does it mean for the plankton community members they most often co-occur with? And we found through our Spearman's rank correlation that for overall Dungeness crab zoea, we saw moderate positive correlations with copepods, crustaceans, and detritus. And this could suggest that zoea are occupying areas with other taxa that might be eating similar things in the trophic community. With the presence of detritus, we know that the microbial loop might be present. We might see higher nutrient recycling. We also know that zoea can supplement their diets with detritus. They can't survive on it alone, but they can eat it. And so potentially further indicating potential trophic interactions as well. We saw that these zoea are occupying different areas across the shelf, and we can see community overlap patterns potentially changing as a result of that. Our early and mid-stage zoea are generally showing the same top important players in our plankton community overlap with crustaceans, copepods, detritus, and so on. But we start to see these Spearman's rank correlations start to decrease with our zoea 4. And we see some shifts in the top players moving into amphipods, for instance. But with zoea 5, we see these correlation strengths really start to drop to near 0, become non-significant, as colored by the gray bins, or even negative. And this shift for these late stage zoea could be indicative of where they are across the shelf. The plankton community might be changing as you move further offshore. It could also show that these late stage zoea might be more independent of the water masses they occupy, with more enhanced swimming behavior. And then lastly, this could also be an artifact of our sampling. Of course, it could be a limitation in the fact that we only sampled so many zoea 5 in our mock nest nets, so this could also reflect that as well. We also saw transect level differences in our in situ imaging. What did we then anticipate to see in our plankton community overlap as a result of that? Generally, again, the top players are still the most important: copepods, detritus, crustaceans, and so on. But this is when we start to see the correlation strength really start to change across any individual transect. And most notably, we saw at Columbia River, these correlation strengths got incredibly strong in comparison to all of the other transects we sampled. And this is indicative that the Columbia River plume is interacting with the ocean front to create a convergent zone that is not only trapping Dungeness crab zoea, but other plankton community members and nutrients, making these co-occurrence patterns much stronger and much more likely at this transect. So in conclusion, the main takeaways of this thesis is that Dungeness crab zoea are experiencing strong interannual variability in their abundance patterns. Zoea are not uniform across space, but they're incredibly patchy and exhibit strong cross-shelf structuring throughout their development. Because of this structuring throughout development across the shelf, this mediates the environment they are exposed to, as well as the community they most often overlap with. Zoea are experiencing conditions that are not matching with our experimental optimal ranges. And coupling in situ imaging with our net sampling improves our ability to resolve biologically meaningful patterns with these zoea. The broader implications of this work are that the recruitment variability that we are observing in these zoeal stages are likely originating in the offshore, preceding the megalopal stage, and potentially explaining the variability we see in the number of returning megalopi to our coastlines. Because of that, when possible, early zoeal dynamics should be incorporated into fishery forecasting and recruitment modeling. These data could help improve future larval transport models as we're able to understand where they are across the shelf and with depth along the NCC. And because of that, by knowing where these zoea are across space, this could help inform what vulnerability they might exhibit to future climate scenarios. Zoea are often considered more vulnerable to the multi-stressor seascape and changes to those environmental conditions. And as a result, what does that mean under these future climate scenarios when the optimal experimental ranges we're using is not matching what they experience in situ? And lastly, when possible, future work should integrate in situ plankton imaging with biological net sampling, because not only are we able to resolve more ecologically important patterns, but we're also able to ask new questions about trophic community overlap and so on. And so I'd like to thank my advisor, Dr. Seuss Bonagel, the members of my committee, Dr. Robert Cowan, Dr. Francis Chen, and Dr. Kim Bernard. I'd like to thank the NOAA Integrative Multi-Stressor Project for funding part of this research. I'd like to thank the plankton ecology members, past and present, including Elena Konzer, Cameron Royer, Luke Bobet, Nick Howard, Marco Corrales-Ugalde, and Kelsey Swaika. I'd like to thank my family and my friends. I'd like to thank the undergraduates who helped sit at the microscope with me and pick out zoea. And of course, I'd like to thank the Dungeness crab and their larvae for being able to support this project. Thank you. Does anyone have any questions online or in person? On Zoom, you are muted. So if you do have a question, please put it in the chat and we will address it. Rick.
Very impressive work for a master's degree. And a really nice presentation also. But my question was, you had mentioned about only about half the zoea that you saw were Dungeness crab. So it seemed like Cancer productus and Oregonensis were probably some of the other ones, but were you able to actually identify them in ISIS down to species?
So that's why we use the proportions. Unfortunately, we are limited in our ability to resolve the species level information. And, as future work, I guess I could create proportions of the diversity of the other crab zoea that we saw across the NCC and then apply that to the ISIS images to see if those, where they occupy across the shelf change. But on average in our mock net sampling, we did see predominance of cankered crab zoea as shown on this right bar plot, where generally it was like 98 to 70% are cankered crab. So it could be another avenue of looking at how these much finer scale resolutions manifest across the diversity of crab taxa that we observed.
Thank you.
Got one in the back, Elena.
Thanks for that great presentation, Meredith. My question is about the different stages of zoea offshore versus inshore. I would expect that pattern that you showed of the later stages being offshore, earlier stages being inshore to be something that might happen during the summer with like upwelling currents pushing I guess, affecting plankton and water offshore, but your data is during the winter.
Yes.
And so is there still currents pushing them offshore a lot during the winter? And is that maybe why they hung around the continental shelf and didn't get like pushed so far offshore? Or is there something else going on there?
So we do have the presence of downwelling predominantly in the winter, which is pushing organisms further onshore onto the shelf than upwelling does. But we do know like there is the advective features. Predominantly, the idea is that these we are advected northward by the Davidson current, and then in the spring transition, when we might start seeing upwelling conditions, they are then advected southward with the California current. So, there are those features here, and I think future avenues of this research should then use these data to improve larval transport models and then address those questions as to what different current features, velocities, and so on might be influencing these transport pathways, because I do believe there are many different mechanisms that could produce these zoea moving further off the shelf, in addition to just being cumulative cross-shelf transport.
We have a question online.
Okay.
Is the variability of zoea distribution a result of time of spawning or sampling, or more so location of spawning? Given that some years had higher density at a specific sampling station and others it was more evenly distributed.
I would say it's a mixture of the two, and we're not necessarily able to tease apart whether it's the source population or when these spawning events occur. We are limited in the data we have available in terms of being able to assess where these zoea might be coming from. And so I would say it's a reflection of when these crabs spawn and where they spawn. And it is a limitation of our study that we are only sampling 1 week of the given pelagic larval duration for these organisms. And so if we did more continuous offshore sampling during the length of the winter season, we might be able to tease that apart better than we are able to here.
All right, this is from Maddie Anderson, I'm guessing a family member.
That's my sister.
You had more time or continued this research in the future, what would you be most curious about to explore further?
I am currently taking Lorenzo Cianelli's fishery oceanography class, and I just learned about the parcel larval transport model, and I think that would be really exciting to learn Python, gain a new skill, and then use this like high resolution larval transport model to like see how these data then reflect where they might end up, where are they coming from. And so research has been done using L-tran larval transport model that was made for oyster larvae, but I'd like to see how the parcel model might do better on that front. Yeah.
Speaking of Lorenzo, he has a question.
Whoa, Lorenzo's here.
Great work, Meredith. Do you think that later zoeal stages can and will vertically migrate?
I do. I think, again, a limitation to our sampling design is that, even within these finer scale resolutions, we are seeing something happening at night. And so I do believe that these late stage zoea can adjust their vertical positioning. It's just a matter of is our sampling capturing that within the length of the transect we sample in each station. And the data shown here are again just for 2019, because our Mezcal cruises sampled day, night, and our Spectra cruises only sampled day for Mach Ness and night in ISIS. And because of that, we might be missing certain behavior features here. So I would say that it might be. Just because it's absent doesn't mean it's not happening. That's Jennifer.
I'll echo what Rick said. That was really incredible and a huge amount of work and a really nice presentation. So this is a really interesting pattern, and it's counter to what people might anticipate in that they're actually descending down during night.
Yeah.
And do you think that that's a seasonal effect? That's maybe kind of a winter versus summer, or?
That's a great question. I can't speak to summer DVM patterns. I don't know if anyone in our lab has looked at diel vertical migration patterns for the species they study. So it could be seasonal in like terms of the phototaxis cues that these zoea might experience in the winter might be different than during the summer. And so maybe light isn't as strong a cue for them to move further to the surface during the winter season. But yeah, I don't know if I can really say one way or another if it's seasonally limited. What was that?
And with this, what we've seen in some other areas, particularly in the Straits of Florida, some more tropical fish, is that you see fairly distinct vertical distributions in the daytime and very diffuse at night. So it's not so much a singular vertical migration one way or the other. It's just we sort of thought it was just the lack of cue, lack of bright light, just enabling everything to spread out. Whether it's a response to light or a response to predators, we don't know. But here you're seeing that diffuse of effect at night, but there's also a little bit deeper, which is pretty interesting. Don't have an answer.
Yeah. And to add to that, in our ISIS data, we actually see this like behavior in the frames we caught in our zoea that they're generally oriented in this manner. And I don't know why but across the board, we had visually confirmed some high abundances that we were observing along Newport Hydrographic in 2023, and we're able to actually see our model was performing well in classifying these zoea correctly, and they were generally oriented in this way across the frames. Awesome. Well, thank you, everyone.
Questions behind?
Yes. Oh, wait, I want my title slide. Okay. I don't want just my little, I mean, that's really cute in the ISIS picture, but. Or can you tell how sweaty I am?
No.
Okay. Okay, thank you. Yeah, I'm curious who showed up out of the bunch. Yes, you honestly could probably end the Zoom if you wanted to. Thank you to everyone on Zoom for coming and listening. I appreciate you all.
Yeah, that's administrative stuff.